we know the zoonosis as psittacosis. Currently, this is a notifiable disease in Australia. Chlamydia psittaci is regarded primarily as a respiratory pathogen, but today I wish to share with you some sentinel incidents regarding the spillover of Chlamydia psittaci from birds to horses and how it did not behave like a respiratory pathogen. In 2014 and 2016, we saw for the first time Chlamydia psittaci causing late-term abortion in pregnant thoroughbred mares and a consequent zoonotic to people handling the affected fetal membranes of the aborted foals. Late-term abortion to Chlamydia psittaci has been reported in literature previously, but very sporadically. However, the 2016 incident resulted in a 20% of cases of foal loss being due to Chlamydia psittaci. Initial work with molecular investigation found the parrot and pigeon strain to be responsible, and this flagged the need to better understand the suspected spillover infection from birds to horses. Chlamydia is a well-known avian respiratory pathogen, so why is it showing a definite preference for the horse now and the reproductive tract and having the clinical sign of abortion? This led to my research questions, which included detecting chlamydia in the pregnant mare, newborn foal, and in birds. A critical question considering that the aborting mare actually gave no sign of the impending loss, maybe a mild vaginal discharge 24 hours beforehand, but that was all. And further questions I asked were, are all pregnant mares affected, or are there risk factors that might focus our attention? For instance, once a mare has aborted, is she then immune? Or does co-infection with another pathogen affect chlamydia? Seeking answers to these questions forms the basis of my PhD. I am currently conducting a 2-year longitudinal study working with 14 stud farms and a group of brood mares across 2 pregnancies, where I am sampling them monthly at 2 anatomical sites and also sampling the newborn foal at birth. I am testing the mare and the newborn foal for Chlamydia psittaci. However, I'm also testing for another pathogen, equine herpes virus 1. This is because equine herpes virus 1 is a respiratory and reproductive pathogen as well and has very similar pathology. However, it is different in that it is very well studied and there is extensive literature on transmission, pathology and management. So it makes it a very useful comparison to the newcomer in the novel host. In addition to sampling the horses, I conduct environmental samples on the paddocks of the study mares looking for Chlamydia psittaci from potential avian reservoirs. The third component is to collect data regarding the brood mares. And this is done to determine the risk factors for the bacteria. The flowchart you can see here describes the collection of samples through the USC laboratory work to determine identity and infectious loads of Chlamydia psittaci and equine herpes virus 1 using species-specific assays and qPCR. Sanger sequencing of the DNA amplicons is used to further characterise the strain and is accomplished using OMPE fragment of the target gene. All of this is done to better understand infection dynamics. I wish to bring you back to the tile called LAMP. This USC laboratory step was ingenious in helping me achieve my goals and was actually developed by one of our own GeneCology group, Martina Uloznik, as part of her diagnostic research. My field research brought in over 500 swabs per month, which is incredibly time intensive and expensive to take it through the normal steps. Using LAMP, which stands for Loop Mediated Isothermal Amplification, I was able to use a raw swab instead of extracted DNA and was able to run the reaction in a significantly reduced time, within 30 minutes. This is due to the use of 6 primers targeting the gene. With this step, I was able to eliminate all negative to C. psittaci samples and take forward to extraction and quantified PCR reaction the positive samples, saving me time and money without sacrificing sensitivity and specificity. So what have I found after 1 year of field research? I'll just discuss with you some work on some answers I have on prevalence, molecular characterisation and risk analysis to date. Chlamydia psittaci, I detected chlamydia psittaci in healthy newborn foals. It was detected at 14.5%, which is actually comparable to equine herpes virus 1, the pathogen which I was using to compare between the two. It was not detected in pregnant mares at any time point tested, and yet the pathogens still pass through the mare to the unborn foal. Looking at the anatomical site detection, variation from nose, vagina, rectum and placenta, it can seem to be highest in the placenta and rectum and lowest in the vagina. The overall site detection rate of 10.3% for chlamydia psittaci indicates that per animal, we were able to detect it at multiple sites than in equine herpes virus 1 at 8.2%. The next piece of the puzzle was provided with molecular characterisation. The image here is of a midpoint rooted phylogenetic tree constructed using alignment of full-length C. psittaci OMP-A sequences representative of the major OMP-A genotypes. The DNA samples were collected from the live healthy foals, environmental avian samples, abortion cases that were reported to the New South Wales DPI for the year, and I included all the historical cases of chlamydia psittaci abortion. You can see in the main clade, you can see, sorry, I haven't got a pointer here. In the main clade, you'll be able to see all the disease-causing strains. Abortion in the horse, psittacosis in humans, in birds, are all clustered and donated with an OMP-A strain 24, or the 6 BC clade. A DNA sample from an Australian white ibis I sampled on a study farm was a distinct genotype F sequence. This suggests that there may be tissue tropism for abortogenic strains based on the clustering of the 6 BC type. Further work is required to determine this. Finally, looking at risk factors for chlamydia psittaci and equine herpes virus 1 detection is done to help identify potential management strategies. The odds ratio is a measure of the association between the exposure and an outcome. So in the column labeled crude is the univariate analysis, where we just look at a factor and it directly relating to pathogen detection. For chlamydia psittaci, we can see that being a maiden is 2.22 times more likely, and being born in winter is 8.33 times more likely, and is statistically significant. In the column next to it, label adjusted, is a multiple logistic regression analysis where we mix up all the factors together to see how that relates to detection. And with chlamydia psittaci, in the final analysis, we can see that the odds ratio is 15.00, and it is statistically significant for chlamydia psittaci. For equine herpes virus 1, we can see that being born in winter actually has an inverse relationship and is statistically significant, being univariate and also in the multiple logistic regression model. So this tells us that being born in spring is much more pertinent to EHV1, equine herpes virus 1 detection, compared to being born in winter for chlamydia psittaci detection. Here is a correlation matrix which just helps describe that big table I had before, where it represents here that the red dots are the positive relationships and the blue dots represent the negative relationships, and the size and intensity of colour are proportional to the correlation coefficients. So we can see here in the model on the left, for chlamydia psittaci, the relationship of being born in winter to chlamydia psittaci, and on the right-hand side, we can see for equine herpes virus 1, we have a negative and a smaller relationship. The future directions of my work, this final year of my study, the finding of chlamydia psittaci in newborn healthy foals is a new finding. Previously, it was only known to be in abortion cases. and this is a very keen area of interest I wish to pursue. Does subclinical levels of chlamydiosis infection affect the development and immunity of the young athlete? But for now, the important implication of subclinical levels of a zoonotic pathogen may affect occ health and safety for all stud workers handling these foals being born. Secondly, understanding the drivers and level of infection load that causes disease is the next step in my PhD. This will inform and lead to improved diagnostics in the pregnant mare and will be achieved by epidemiological statistical analysis. Finally, the new knowledge of the focus on wintertime allows for creative management strategies to be considered regarding the interactions of the birds and horses and may assist in preventing foal loss and zoonosis. Finally, there's a number of people to thank, and I will be thanking these people for the next few years, I'm sure. They're all listed there, and I wish to acknowledge them all. Thank you for your time.
Thank you very much for a fascinating talk. I was wondering if, now that you know that the chlamydia seem to be related with the abortion, is that something like an antibiotic treatment is something that they could consider?
It's very interesting because there's a couple of ways to approach it. As we're dealing with an infection, a potential suspected spillover from birds, we could be looking at more an ecological than, say, a traditional intervention with a vaccine. Still more work needs to be done to understand why the drivers to infection as regards to, is it just certain times that we need to be aware of this, or is this something that requires a vaccination? Current medical therapies with tetracyclines and vaccinations for chlamydia are about, but certainly not for equine, and there are consequences when you're dealing with the foal, the unborn foal.
Okay, we have a number of questions here from, and congratulations to you, Susan, but we'll go to the questions. Sam is asking if you can get samples from the different anatomical structures of the placenta to determine transmission from mare to foal.
Histological work is underway, complementary to my work with another group. It's still not determined the exact method that it enters the placenta. I sampled both nasal and vaginal samples and was not able to statistically differentiate the mode that it actually is arriving to the uterus. And we need more work in that area because that is an important fact. And with regards to the placenta, I just had general samples. I wasn't able to, I wouldn't be able to determine if it, at this stage, that answer.
And there's another question, if you think it's possible transmission to exposed humans.
Birds can actually infect humans. It does occur. As I was saying, it's a notifiable disease. It's a direct transmission from birds to humans. And in 2014 and 2016, we had where the birds infected the horses, and then the horses' membranes went on and passed it on to the humans working. One health approach has been applied to it, and it is considered a low risk, where you need to be in an environment where you have a very high contact and high exposure, and understanding it only happens at certain times. So there is an awareness there that needs to be further investigated. But it was checked out from the human point of view and was considered a low risk.
All right, thank you very much. I really appreciate your talk.